Now that we have been through the small intestine, let's take a quick detour by the pancreas. So the pancreas is a very important accessory digestive organ that's going to secrete enzymes and buffers into the duodenum and will continue the work of digestion that was begun in the mouth and in the stomach. The regions of the pancreas include this head region here, which is tucked into the loop of the duodenum, and then we have the body of the pancreas and then the tail of the pancreas. And you'll see that there are these lobules, and these will be segregated by some connective tissue septa, and septum means wall, and that you're going to have some connective tissue separations, partitions, that are going to separate these lobules, and the lobules are full of pancreatic acini, or tubuloalveolar glands that are going to end in these pancreatic acini, which are these blind sacs. And these are going to be lined with a simple cuboidal epithelium. And these are going to produce our enzymes and our secretions of the pancreas, which will include enzymes, buffers, ions, water. And we end up making about 1 liter of pancreatic juice per day. And that pancreatic juice will then be dumped into the duodenum via the pancreatic duct. And the pancreatic duct will empty first into this duodenal ampulla right about here, which will also be receiving the common bile duct. And the common bile duct is coming from emergence of ducts, the hepatic duct from the liver and the cystic duct from the gallbladder, and they will converge and form the common the common bile duct. And that will also empty into this duodenal ampulla, and then you'll have your hepatopancreatic sphincter right here, which, as you recall, under the influence of cholecystokinin that will be released by the enteroendocrine cells of the duodenum, that will cause that hepatopancreatic sphincter to open up, relax, and allow the contents, both the bile and the pancreatic juice, to come into the duodenum. And sometimes these structures have their eponyms. For example, this is the duct of Wirsung, and then we have the ampulla of Vater, and of course we also have the sphincter of Addi. So they all have their eponyms. It's probably a little bit easier just to remember them by their descriptive names. But if you hear those terms, then that's just simply some of the eponyms, some of the older terms that are still in use. Now, about 3 to 10% of the population have this accessory pancreatic duct. Not everybody has this, but I mention it as a side because the book points it out. It doesn't really serve any particular function. It's just another route for pancreatic juice to get into the duodenum. Now, when we look at the lobules of the pancreas, remember they're separated by these partitions called septa. And they're going to have not only blood vessels and tributaries of the pancreatic duct, but each lobule will have a bunch of tubuloalveolar ducts or glands that have repeatedly branched and end in these blind pockets called pancreatic acini. So if we kind of zoom in on them, here's a cartoon version of them. Here we can see the pancreatic duct. Here we can see some tributary ducts branching off, and we're going to have these pancreatic acini, or these blind sacs. And they are going to be lined with this simple cuboidal epithelium here. Now, scattered throughout the exocrine part of the pancreas, and the exocrine portion is these pancreatic acini, we're going to have the pancreatic islets of Langerhans. And remember, these are accounting for only 1 to 2% of the pancreas, and these are going to be secreting our endocrine hormones such as glucagon and insulin. But remember that the exocrine function of the pancreas takes up about 98% of the actual physical part of the pancreas. In terms of the endocrine function, while extremely important, and you can't live without it, the 98% of the pancreas is actually dedicated physically to the exocrine cells. And as we've said, this is going to be producing about 1 liter of pancreatic juice per day, which once again is going to be a mixture of enzymes, buffers, for example, bicarbonate buffers, phosphate buffers, ions, water, and our pancreatic enzymes. So all of this mixed together is called pancreatic juice. And that pancreatic juice will be about 1 liter a day being produced, and it's all going to be under control of hormones from the duodenum, as well as the parasympathetic nervous system. So even during the cephalic phase of digestion, when the stomach is preparing to receive the food, the pancreas also has to start producing enzymes because it takes a long time actually to make enzymes. So by the time you're thinking about food, your pancreas is already starting to make the enzymes that it will need when the food arrives into the duodenum. And it takes a bit longer to make the enzymes than it does to make the buffers. So these enzymes will be started upon by the time you put the food in your mouth. All right, so let's talk about some of the pancreatic enzymes. Just like we had amylase made in the mouth, we have a pancreatic alpha amylase that is very similar in composition that will break down starches. We have a pancreatic lipase, which will break down will break down lipids into fatty acids. So say you have a triglyceride, you're going to be pulling off the glycerol molecule and yanking the fatty acids off of that. We also have nucleases, which are going to break down our nucleic acids, as well as proteolytic enzymes. And as the name implies, protein, lytic, breaking, these enzymes are going to break proteins. Then we have two types of enzymes, really. We have the peptidases, which are going to break small polypeptides into their amino acids. So as the name implies, peptidases are going to break peptide bonds, and proteases are going to break down the larger protein complexes. So this is going to be important because, once again, between these enzymes, our pancreatic alpha amylase, pancreatic lipase, and our proteolytic enzymes, we're going to continue the work of digestion that was started in the mouth and the stomach. And we're also going to be breaking down RNA and DNA and that kind of thing as well. So some of the proteolytic enzymes that we're going to see are going to be things like trypsinogen, chymotrypsinogen, procarboxypeptidase, and so forth. And these are going to be, as in the stomach, secreted as proenzymes, and then later they will be converted into their active form. So trypsinogen will be converted into trypsin. And these will attack very specific peptide bonds. So these peptidases will break down very specific amino acid bonds between specific amino acids, I should say. And so a lot of these things are going to be activated, I should say, by some of the brush border enzymes. So when they get into the lumen of the duodenum, they'll be activated by our enteropeptidase that we talked about a little bit earlier.